Dear students, I am Miss Prabhavati Essen. You are aware of the fact that our body needs various kinds of macro as well as micronutrients for sustaining the life. One such nutrient is vitamin E, which plays a wide variety of roles in various biological systems. Vitamin E is basically an antioxidant nutrient. Today I will be talking to you about the history, evolution and various aspects related to vitamin E. After studying this module, you would be able to understand the discovery of the nutrient, various forms in which it exists, mode of absorption, major food sources, health benefits and effects of deficiency. Now let us get to know the history of vitamin E. Vitamin E was basically discovered and characterized as a fat-soluble nutritional factor essential for normal reproductive processes. This specific role of vitamin E was found out in the course of carrying out reproductive studies with rats. In the year 1922, it was demonstrated that to prevent fetal death and sterility in rats, an important fat-soluble dietary constituent was identified to play a vital role in this regard. Initially, this was termed as factor X and was also being referred to as anti-sterility factor. Later, the vitamin E was assigned. During the year 1936, it was isolated from wheat germ oil, which was termed as tocopherol, which is derived from the Greek word tokos and pherin. Several different forms of tocopherol have been identified till date. The term vitamin E is often used to denote a mixture of biologically active tocopherols. In general, there are 8 naturally occurring vitamin E compounds which are mainly synthesized by plants. Vitamin E is a general term that encompasses a group of highly potent, naturally occurring lipid-soluble and chain-breaking antioxidant substance. There are totally 4 tocopherol compounds such as alpha, beta, gamma and delta tocopherol. About 4 types of tocotrienols have also been discovered. Alpha tocopherol is the most abundantly available in nature and has been shown to possess highest biological activity and also has a vital role in reversing vitamin E deficiency symptoms among humans. In the year 1950s, it was rediscovered that vitamin E to have a specific role in cellular antioxidant system along with sulfur amino acid and selenium. Subsequently, it was also proven that it is very effective in preventing lipid peroxidation and various other events associated with radical-driven oxidative stress. Now coming to its structure, the basic structural unit consists of a chromin ring system along with an isoprenoid side chain having 16 carbon atoms, and this is common to all homologs of vitamin E. The major vitamin E homologs, namely alpha, beta, gamma and delta, differ in number and position of the methyl substitutes in the basic chromin ring. Tocopherol differs from tocotrienol since it possesses a saturated side chain. In order to perform the antioxidant activity, tocochromonols must contain a phenolic hydroxyl group. Most importantly, it should have one methyl group in the benzene ring. Since alpha tocopherol contains 3 methyl groups, it is considered to be the most active of all the homologs. The major forms which are retained in human plasma are the 3R alpha tocopherol and 2R stereoisomers, RSR-RSR and RSS alpha tocopherol and various 2S stereoisomers respectively. Vitamin E activity is basically expressed in terms of 3R alpha tocopherol equivalents, which accounts for about 90% of the activity in human tissues. 1 international unit is equal to 1 milligram of alpha tocopherol acetate. Now let us look into the major food sources. Vitamin E is synthesized only by plants and hence is primarily found only in plant-based foods. Thus, fats and oils of vegetable origin are known to be the richest sources of vitamin E. Vegetable oils and oils derived from food grains have been estimated to contain about 50 to 100 milligram per 100 gram of the product. Only 8 milligram is present in coconut oil. Wheat germ oil is exclusively the rich source of vitamin E, containing about 260 milligram per 100 gram. The chief sources in Indian diets are the vegetable oils, nuts, oil seeds, and whole grains. Significant amount of tocotrienols are found in palm oil, rice bran oil, and the bran and germ portions of certain cereals like oats, barley, and rice. Vegetable oils are generally subjected for the process of refining. This would result in a loss of significant amounts of vitamin E due to the effect of processing conditions. The Western diets are estimated to contain 15 milligram of vitamin E. The Indian diets are also found to contain almost similar amounts. On an average, consuming 20 gram oil would contribute 10 milligram of vitamin E. Hence, consuming 400 gram of food grains would have been found to contain 6 to 8 gram of fat, out of which 3 to 4 gram of vitamin E might be obtained. In case of Tamil Nadu, the major staple cereal used would be the rice, and it is a well-known fact that rice contains very minor quantities of vitamin E. The oil consumption was also determined to be between 5 to 10 gram per day. Such a diet would provide less than 5 milligram of vitamin E. The demonstrated effects of vitamin E deficiency would suggest that it is desirable to consume adequate quantities of whole grains and oil seeds. Leafy vegetables also contain around 0.5 to 1.5 gram fat per 100 gram, and this is thought to yield 0.5 to 1.5 milligram of vitamin E. Okay. Now, having known about the major food sources, let us know about its absorption. Generally, the efficiency of absorption of vitamin E is low in humans. The absorption of free alcohol forms of tocopherol and their esters are highly dependent on adequate pancreatic function, biliary secretion, micellar formation, uptake into enterocytes, and chylomicron secretion. Presence of any defects at these sites could impair the absorption process. The primary site of absorption is the small intestine, which occurs via micelle-dependent diffusion. Vitamin E absorption is estimated to be highly variable and can range from 20 to 70%. During the initial stages of absorption, emulsification takes place in the stomach and later in the small intestine in the presence of both pancreatic and biliary secretions. This results in the production of mixed micelle molecular aggregates, which then solubilizes tocopherol and transport it to the brush border membrane of the enterocyte through passive diffusion. The cellular uptake of vitamin E can occur through two main modes: as a receptor-mediated process in which the vitamin E gets delivered by the liver into the cells. The other process is mediated by lipoprotein lipase. The tocopherol esters, which might be present in processed foods and vitamin supplements, needs to encompass a hydrolysis step in small intestine prior to absorption. The transportation of vitamin E in the blood mainly occurs through plasma lipoproteins and erythrocytes. Chylomicrons facilitate the transportation of tocopherol into the systemic circulation, wherein they would be catabolized by lipoprotein lipase to its subsequent chylomicron remnants. This further transports tocopherol to high-density lipoprotein. This tocopherol in HDL would be further transferred to other circulating lipoproteins, that is low-density lipoprotein and very low-density lipoprotein. This helps to protect the lipoproteins from oxidation. The level of vitamin E in blood is dependent to a greater extent on the degree of VLDL secreted by the liver. The alpha tocopherol then enters the cells of peripheral tissues within the intact lipoproteins through the LDL receptor pathway, though considerable amount may be taken up by membrane binding sites also. Though the process of absorption of all the tocopherol homologs in the diet is similar, it has been observed that the alpha form predominates in blood and tissues. This could be due to the preferential selection of alpha form over other forms by the binding proteins. In most of the non-adipose cells, vitamin E is found to be located almost exclusively in membranes from which they can be readily mobilized. In case of adipose tissues, it is partitioned in the bulk lipid phase and it cannot be readily mobilized. The rate of metabolism of vitamin E is said to be limited. Primarily, it gets oxidized into the most biologically inactive tocopherol quinine, which is further reduced to tocopheryl hydroquinone. The conjugates of glucuronic acids of the hydroquinone are secreted in the bile. Other oxidation products, including dimers and trimers, as well as adducts, are formed during in vitro oxidation. The main mode of excretion is through urine. With increasing doses of supplemental vitamin E intake, a consequent increase in urinary excretion of the alpha carboxyethyl 6-hydroxychromone metabolite tends to increase. The excretion of this metabolite is estimated to be 3 times higher than those of alpha tocopherol and 3R alpha tocopherol, whereas the concentration of 3R alpha tocopherol is considered to be much higher in plasma. This suggests that these urinary metabolic products are the major indicators of non-utilized forms of vitamin E. Of the total amount of ingested alpha tocopherol, half of it would be metabolized and predominantly excreted as alpha CEHC. This metabolite has been proposed to inhibit potassium channel and has a tendency to increase the urinary sodium excretion. Fecal excretion is the major route of excretion as intestinal absorption is relatively low. Excess intake of alpha tocopherol as well as other forms of vitamin E which are not preferentially used would get eliminated in an unchanged form in bile. Storage. The tissue uptake of vitamin E mainly takes place through plasma. The tissue uptake of vitamin E involves several major routes which are as follows. The catabolism of triglyceride-rich lipoprotein by lipoprotein lipase, the uptake of low-density lipoprotein through LDL receptor, delivery systems facilitated by high-density lipoprotein, non-specific transfers taking place between lipoproteins and tissues. Vitamin E gets rapidly transferred between various lipoproteins and also between lipoproteins and various membranes which may have a role in enriching membranes with vitamin E. The human plasma phospholipid transfer protein is known to be involved in accelerating this process. Coming to the major functions, antioxidant activity, tocochromonols are one of the most effective group of lipophilic phenolic antioxidants. Researchers have proposed that antioxidants protect key cell components by neutralizing free radicals before they can cause lipid oxidation or DNA damage. By reducing free radical attack, antioxidants help to break the chain reaction of lipid peroxidation and they protect the cell membranes by facilitating the processes of lipid repair and lipid replacement. Through this mechanism, they may prevent cancer or heart disease or any other form of degenerative disorders. Epidemiological studies have indicated that diet-derived antioxidants such as vitamin A, C, and E may be important in maintaining human and animal health since they play an essential role in quenching the free radicals. Accumulation of free radicals is known to be a major factor leading to the occurrence of several lifestyle-related disorders. Hence, for maintaining long-term health, it is necessary to include adequate amount of antioxidant-rich foods in our daily diet. It has been evidenced through epidemiological studies that high plasma concentrations of vitamin E are associated with a low risk of cardiovascular disease and certain types of cancer. The effect of a mixture of alpha, beta, gamma and delta tocotrienols and alpha tocopherols DNA damage was determined in a randomized double-blind placebo-controlled study. DNA is usually considered a target compound in oxidation process. This revealed that supplementation with vitamin E mixture reduced the level of DNA damage among healthy subjects. This observation has indicated to have a possible relationship between the molecular mechanisms involved in formation and repair of DNA. Role in cardiovascular disease. Supplementary vitamin E was reported to be effective in reducing atherosclerosis progression in subjects with previous coronary artery bypass graft surgery who have not been treated with lipid-lowering drugs. Tocotrienols in animal cells inhibit cholesterol biosynthesis by suppressing 3-hydroxy-3-methyl-glutaryl-CoA reductase enzyme, which is a key enzyme in the sterologenic pathway. This results in reduced manufacturing of cholesterol by liver cells. Role in cancer. Evidences regarding the anti-proliferative and apoptotic activities of tocopherol on normal and cancer human cells are available. The mechanism could be related to induction of apoptotic phase mainly via a process mediated through mitochondrial pathway and cell cycle arrest, which happens due to the suppression of cyclin D by tocotrienols. Tocotrienols also inhibit vascularization, reducing proliferation, and malignant proliferation demands elevation of HMG-CoA reductase activity, and tocotrienols suppresses its activity. Only few clinical trials were carried out to determine the effects of tocotrienols on cancer prevention or treatment. Tocopherols can be used in order to reduce the potential toxicity due to intake of 13-cis-retinoic acid in subjects at high risk for lung cancer. Exposure to tobacco smoke is a major cause of lung cancer. About 50% of new lung cancers arise in former smokers, and effective chemoprevention strategies are critical to reduce the risk. Chemoprevention is an emerging field whereby drug therapy is inhibited to halt or reverse the carcinogenesis process before the emergence of invasive cancer. Based on epidemiological data, it has been demonstrated that lung cancer patients have lower serum levels of several antioxidant vitamins. Other related functions. Apart from its antioxidative role, vitamin E takes part in other biological processes also, which are as follows. Facilitates the maintenance of cell membrane integrity, exerts anti-inflammatory effect mainly by direct and regulatory interaction with the prostaglandin synthetase and complex of enzymes which are mainly involved in the metabolism of arachidonic acid. It also plays a major role in DNA synthesis. It stimulates immune response, regulates intercellular signaling and cell proliferation mainly by modulating protein kinase C activity. Now let us know about the adverse effects of vitamin E deficiency. Vitamin E deficiency is rarely encountered in humans. Overt deficiency symptoms in normal, healthy individuals consuming diets low in vitamin E have never been described. Vitamin E deficiency may occur only as a result of certain genetic abnormalities occurring in alpha tocopherol transfer protein. In case of various fat malabsorption syndromes or in severe protein energy malnutrition, insufficient dietary intake of the vitamin is the common cause of deficiency. Several other factors have also been implicated to affect the need for vitamin E. Both selenium and polyunsaturated fatty acids are considered to be the most important factors with regard to affecting the vitamin E intake. Selenium tends to spare the need for vitamin E, and hence adequate intake of vitamin E becomes even more important for individuals consuming low selenium diets. The deficiency symptom is primarily manifested as peripheral neuropathy, which is characterized by the degeneration of the axons in the sensory neurons. Other manifestations in humans include spinocerebellar ataxia, skeletal myopathy, pigmented retinopathy, increased erythrocyte fragility, and increased production of ethane and pentane. In experimental animals, the induced deficiency of vitamin E produced various symptoms including hemolysis of RBC, which occurs due to changes in the cell membranes, muscular dystrophy, and infertility among males, and among females unsuccessful pregnancies have been noted since its deficiency results in the failure of the fetuses to reach full term. Severe forms of vitamin E deficiency is known to affect reproductive performance, and the condition has not been demonstrated among humans. But yet, the deficiency increases the tendency for the RBCs to hemolyze, leading to hemorrhage, and death have been observed in some newborns and premature infants, and is thought to be directly associated with low levels of blood vitamin E. Among malnourished children, the blood levels of vitamin E have been found to be relatively low since they have poor intake or poor absorption. Hemolytic anemia resulting due to vitamin E deficiency is being reported in some of the Middle Eastern countries also. Toxicity. At present, there are no reports regarding the adverse effects resulting due to the consumption of vitamin E from natural foods. It is one of the least toxic vitamin. Both humans as well as animals are capable of tolerating relatively high intake levels of more than 100 times the estimated requirement. At very higher doses, vitamin E is thought to impair the utilization of the fat-soluble vitamins in the body. For instance, animals that were supplemented with high doses of vitamin E were shown to have developed impaired bone mineralization, impaired storage of hepatic vitamin A, and prolonged blood coagulation. Now let us learn about the factors that will affect vitamin E requirement. The most important factor affecting vitamin E requirement is the bioavailability. The dietary vitamin E is found in food that contains fat. For the optimal intestinal absorption, it requires micelle formation and chylomicron secretion by the intestine. Any impairment in the secretion of the absorption facilitating agent would affect the bioavailability. The requirement for vitamin E would tend to increase when the intakes of polyunsaturated fatty acids increases. The suggested ration is at least 0.4 mg alpha tocopherol per gram of polyunsaturated fatty acid should be consumed by adults. It is also been reported that the requirement for vitamin E increases along with increasing body weight. Requirement. There are very limited data on vitamin E requirements since it occurs most widely in vegetable oils and plant foods, and dietary deficiency of this vitamin is not normally encountered. Alpha tocopherol content of vegetable oils and invisible fat in cereals and other foods is generally adequate to meet the daily requirement. Requirement is limited to essential fatty acids. Alpha tocopherol requirement is related to its major role in protecting antioxidant property of essential fatty acid content in the diet, and the suggested intake is 0.8 mg per gram of essential fatty acid. This roughly contributes about 8 to 10 mg tocopherol per day depending on the type of edible oil used. Vegetable oils and invisible fat of cereals and other foods like nuts and vegetables contribute to adequate tocopherol content in Indian diets. Now let us summarize today's episode. Vitamin E is basically an antioxidant nutrient which often denotes a mixture of biologically active tocopherols. It is abundantly available in nature. It is synthesized only by plants and hence is present only in plant-based foods. Vegetable oils and oils derived from food grains are known to be the good sources of vitamin E. Wheat germ oil is exclusively rich in vitamin E. It is known to function as an antioxidant which helps to prevent the occurrence of cancer and cardiovascular disorders. Deficiency is rarely encountered among humans, which might occur as a result of genetic abnormalities. Thus, in order to obtain optimal health benefits, it is essential that the individuals must consume them in adequate amounts for maintaining health and longevity.